we keep looking at this DNA stuff, but did you ever wonder, what does it really do? I mean, what does it actually do? Well, here we are with making protein! Or, what's DNA really do? It's me, Moser. And after watching this video, you should be able to identify protein synthesis as the process that is used by cells to construct proteins. You should also be able to describe proteins as chains of amino acids held together with peptide bonds. And you should be able to name and describe the steps, structures, and molecules used in protein synthesis. It'd be really awesome if you already knew what DNA was, and you should know your base pairs, and where DNA is in cells. Okay, let's get started. Okay, so, DNA, we've looked at it, we've seen how it replicates, we've unzipped it, we've zipped it up, we've done all this stuff, but I got questions. How can a collection of chemicals, that's all it is, be instructions? How can this be a blueprint for anything? It's a chemical. DNA actually does contain instructions, and it contains instructions, kind of like a WikiHow site, for building proteins. Proteins are long molecules. They're made up of smaller building blocks called amino acids. And that would be these little guys. The amino acids are joined by peptide bonds into long chains, and then they're folded. And they are folded in strange and amazing ways. Seriously. The process of building proteins is called protein synthesis. To synthesize is just to build something, like photosynthesis builds things out of light, and protein synthesis builds proteins. Remember, proteins are just chains of amino acids held together with peptide bonds and then folded like crazy. Now, all organisms, from prokaryotes to eukaryotes, all organisms construct protein. From bacteria to blue whales, from earthworms to ferns, from maple trees to crayfish. Every single organism makes proteins. But why? Well, proteins are pretty important. They make up muscles and skin. They provide structure for cells, like bacterial cells and plant cells. The cell wall is made of protein. They act as chemical messengers in the body. Proteins also build enzymes. We've talked about those a few times. Those funny little chemicals that help other processes run inside living things. Now, proteins, as you remember, are built out at the ribosomes, and all organisms have ribosomes in their cells. But in eukaryotes, the DNA, that's the instructions for making the proteins, is kept in the nucleus of the cell. DNA is a really long molecule, and there's a lot of DNA in there, and only parts of it need to be read at any one time. A gene is a section of a strand of DNA, like that little orange highlighted section. The puzzle is, how do you get a long, tangly, stringy molecule like that out of the nucleus to build the protein for just a single gene without creating a hopeless mess? Oh, that's easy. You can't. Oh. Since only a tiny little short section of DNA needs to be read at any time and have the proteins built from it, there's a different way to do this. Instead of dragging the whole DNA molecule out of the nucleus, a short section, a little section of the DNA is unzipped. This sounds like replication, but that's where the similarity stops. A copy is made of 1/2 of the DNA molecule. This copy is made out of another nucleic acid, RNA, and this RNA is mRNA, to be specific. RNA is different from DNA in a few important ways. For one, RNA is only one-sided, so it kind of looks like half of a DNA molecule. Also, RNA doesn't have any thymine. There is no thymine in RNA. In its place, we find a base called uracil. Messenger RNA, or mRNA, is the molecule that gets copied from DNA. The process of making a copy of DNA in RNA is called transcription. The cell is transcribing or copying the information from one nucleic acid, DNA, to another, mRNA. It's the same language, the language of nucleic acids, but in a different form. This is like taking a large book, maybe a cookbook, and copying a short section of text onto a little note card. Much more portable. This little copy of the information from the DNA can now leave the nucleus through a nuclear pore. These are the little holes that cover the surface of the nucleus. And once it leaves the nucleus, it can travel all the way through the cytoplasm out to the ribosome. Yay! Now what? Once mRNA gets to the ribosome, it gets fed through it and is read by the ribosome. The mRNA is read in 3 base chunks called codons, and each codon actually codes for a specific amino acid. As a codon is read, an amino acid is delivered to the ribosome, carried on a small molecule with a code, called an anticodon, that complements the codon. The codon provides the order for the amino acid, and the anticodon, well, it's on the tRNA, but it's really just like a license plate on the truck that delivers the amino acid. In other words, the anticodon only provides a confirmation that the correct amino acid is delivered. As the next codon is read, the next amino acid gets attached to the previous amino acid with a peptide bond. This keeps on going until the ribosome reads a stop codon. The process of creating a chain of amino acids from mRNA is called translation. Now the cell is translating the information from the language of nucleic acids to the language of proteins. This is just like if you take a word in one language and use a dictionary to translate it into a different language. Once it's complete, the amino acid chain is released, and it's sent off to be folded correctly in order to perform its job. The mRNA will be recycled, and the ribosome is ready to accept a new piece of code. Once that amino acid chain, or polypeptide chain, gets folded, and man does it get folded, it's now a protein. Okay, protein synthesis in 10 seconds or less. DNA is a nucleic acid. It gets transcribed over to RNA, which is a nucleic acid, and RNA is translated into a chain of amino acids, which get folded to make proteins. Ta-da! You got questions? Well, let's run down your learning objectives. Can you correctly identify protein synthesis as the process that cells use to construct proteins? Well, I hope so. Can you describe proteins as chains of amino acids held together by peptide bonds? Again, I should hope so. And can you name and describe the steps, structures, and molecules used in protein synthesis? Well, I certainly hope so. If you still need help or you still have questions, ask them and keep working. Bye.